Thank you very much. It is my greatest pleasure to be here and to talk about short story of EPA in Japan. Today, I talked two issues in my presentation. The first is a story of field work in Chiba Prefecture. And second issue, how we Japanese people developed a new drug of highly purified EPA and to use as anti-atherosclerotic disease in Japan. This slide shows the step of EPA research in Japan. I deeply that by talk by Spector, he showed the very detailed story of prostaglandin research in the world. 1977, our Chiba University started the prostaglandin research, and then 2 years later, 1979, we started EPA project. Same time, the start of prostaglandin research in the world. At that time, two major problems is these. One is determination of EPA and other fatty acids in human blood using gas chromatography. And second problem is evaluation of human hemostasis, mainly using platelet aggregation. We established this basic technology and then start the field work in the Chiba Prefecture, comparison between fishing village and farming village. Under the collaboration with Chiba Prefecture. And then we select two villages. One is located on sea coast of Chiba Prefecture, and one farming village located in central part of Chiba Prefecture. And then finally, we started first field study in fishing and farming villages in Chiba Prefecture. I remember this year, 3 researchers get Nobel Prize due to the discovery of prostaglandin, thromboxane, and leucotrienes. And then talk about background of EPA study in Chiba Prefecture at that time. At 1970s, aspirin was only one anti-thrombotic agent available in clinical use. No other choice. The number of the patients suffering from thrombotic cardiovascular disease, brain stroke, or myocardial infarction was increasing also in Japan. The research and development of new anti-thrombotic agents was an emerging challenge in Japan. Also, as shown by Professor Spector, a marked progress in prostaglandin and thromboxane research, including possible anti-thrombotic action of EPA at end of 1970. This slide shows the location of two villages in Chiba Prefecture. One is farming village named Kamagaya, and another is fishing village Kawas. This is a landscape of a fishing village. The aim of field study in Chiba Prefecture is as follows. One is to clarify daily intake of dietary EPA in fishing and farming villages. Second, to clarify plasma fatty acid profile, especially the ratio of EPA to AAA in fishing and farming villages. And third, to clarify difference in hemostatic function, especially platelet aggregability, determined as threshold dose of ADP in fishing and farming villages. This picture is how we performed field study in the fishing and farming village. These staff measuring platelet aggregability in fishing village. This is a machine to determine the threshold dose of ADP in these people. This slide shows a summary and a copy from the paper published in Lancet, 1980. The summary first is the daily intake of EPA is in fishing village 2.7 grams per day. In farming village 0.9 grams per day. The difference is 1.8 grams per day. This is later the daily dose of EPA in Japan. And second, the ratio of EPA to AA is 0.58 in fishing and 0.41 in farming. And finally, the platelet aggregability determined as threshold dose of ADP is 6.6 micromole in fishing and 2.3 in farming. This slide shows a summary of fieldwork study performed in 35 years ago in Chiba Prefecture. Daily intake of EPA in fishing was 2.7, and that of farming is 0.9. The difference is 1.8. 1.8 grams per day is very important in Japan. And second, plasma EPA AA ratio is 0.56 in fishing and 0.41 in farming. Platelet aggregability determined as threshold dose of ADP is almost 3 times higher than that in farming village. This means the platelet aggregability in fishing village is markedly suppressed by taking so much EPA. At this time, I show you 2 persons to whom our EPA study in Japan is deeply indebted. Dr. Yon Dalbel. This picture is 35 years ago. He was invited in Japan. Also, second person is Sir John Bain, you know, Nobel Prize of 1980. This photo is me, 35 years ago. I'm still now younger. Today, the second issue depends on these findings in field work study in Chiba Prefecture, how we develop a new drug of EPA, three legacy of early study in Japan. First is new drug, highly purified EPA. The second, to which patient? Third, how much? That means daily dose of EPA. To develop a new drug, that means highly purified EPA by Nisui. This one poster coupled EPA group of Chiba University to Nisui company because I was young medical doctor at the time. I see this poster, railway station, big surprise. I believe this company make highly purified EPA under the collaboration with us. And then I visit the company of Nisui at Tokyo and start this story. You know? You believe? This slide shows the flow chart of purification of EPA from fish oil. So complicated. I don't know so in detail. Second, I emphasize during the purification of EPA from marine oil, it's very important to recognize some compounds. This means that to neglect dioxin from marine lipids. This process markedly decreases the content of dioxin in final product of EPA. Some laboratories compare the different type of highly purified EPA, generic for different type, and EPDEL, developed by Nisui. The content of dioxin is EPA by Nisui is very low of dioxin. This process shows the second and third step to increase the content of EPA from 30% to 80%. And finally, the purity of EPA is 98, almost 100%. This shows the gas-chromatographic chart of crude fish oil and final product of EPA. The purity is 18% to 98%. This highly purified EPA is manufactured by two plants of Nisui. One is Kashima plant, and second is a Tsukuba plant. And then go to next issue. To whom EPA is administered? Answer: patient with low EPA AL ratio. This slide shows the result of field work study performed in Hisayama town, located in Kyushu Prefecture, almost 10 years ago, performed by Kyushu University. They determined the rate of ratio of EPA and AA in the resident of this town. Left side shows the median of EPA AA ratio in Hisayama village is 0.5. That is same, just same, the value in farming village observed 35 years ago in Chiba Prefecture, same value. After this, in Japan, to measure EPA-AL ratio in the patient with atherosclerotic disorders are supported by medical insurance. It takes 4,000 yen paid by patient is almost 1/3, 1,500 yen. Sample is serum or plasma. This measuring is performed by several companies, SRL, BML, and the method is gas chromatography. The indication of medical insurance for this measuring is myocardial infarction, brain thrombosis, ASO, and coronary artery disease. Duration from the sampling to the report is only 3 to 5 days. This slide shows the report of EPA-AL ratio from the company to hospital. This patient showed EPA-AL ratio is only 0.25. This slide shows the result of fieldwork study performed in Hisayama town in Kyushu Prefecture. You can see in the right side of slide the people having high sensitive CRP over 1.0 people having EPA-AL ratio less than 0.41, Q1 and Q2, significantly higher instance of cardiovascular disorders. This result is presented at American Heart Association several years ago. And due to these findings in Japan, patients with cardiovascular disorders whose EPA-AL ratio less than 0.4 are recommended to take EPA. And third issue, how much of EPA be administered? Very difficult question. Answer is, daily dose of EPA in Japan is 1.8 gram per day. How we determine this daily dose? I show you the first paper describing the daily dose of EPA in Japan. Reported by Goto and Kumagai. A clinical pharmacological trial of icosapentaenoate, highly purified EPA, to determine its daily dose. This slide shows the result of these clinical trials. 3 different daily doses of EPA was selected. 900, and 1,800, and 2,700 milligram per day for each. And finally, the observation period is 16 weeks. We checked the change of serum cholesterol. In this slide, daily dose of 1.8 gram or 2.7 gram is no difference. And second, we check the effect of these three different daily dose EPA on the level of serum triglyceride. Baseline is over 150 milligram per deciliter. Finally, both of 1.8 gram and 2.7 gram is similarly suppress, decrease the triglyceride around 70%. However, 0.9 gram, no significant effect. Also, we check the change of the ratio of EPA to AA in these patients. Dose response increase of EPA-AA ratio was observed between 0.9 gram per day and 1.8 gram per day. However, there is no such dose-dependent increase in EPA-AA ratio between 0.8 and 2.7 gram per day. And finally, daily dose of EPA was fixed as 1.8 gram per day in Japan. I already mentioned this value is the same of the difference of daily take of EPA in fishing and farming village in Japan. This slide shows the change of market volume of highly purified EPA as a prescription drug in Japan from 1999 to last year. I can say three important points in this slide. One is approval by the Japanese government as a prescription drug at 1994, sorry. Medical effect is ASO, and second is hyperlipidemia, mainly decreasing triglyceride. Second point is JERRY study. This study is a combination of statin and EPA have beneficial effect on cardiovascular disease in Japanese. Due to these two evidence, finally, 6 years ago, three major Japanese medical associations recommend the use of EPA to atherosclerotic or thrombotic disorders. One is Atherosclerosis Society of Japan, and second is Japanese Circulation Society, and third is the Japanese Stroke Society. And finally, the market of EPA, highly purified EPA, in Japan is increasing from only 40 tons to 500 tons, almost 10 times high increase in Japan. However, concerning about supplements containing EPA and DHA, this slide shows a comparison in Japan and the United States. In Japan, shown in red color, is no significant increase during past 10 years, while United States, starting point, same value. But at this time, at this point, almost 10 times increase. What is the difference? I can say there is next possibility, so many companies to increase market in Japan. I understand step-by-step development of our approach to produce new drug in Japan and almost 10 times increase of the market in clinical medicine. However, in healthcare field, no such progress in Japan. I can say the next problem. Okay. Also, I finally add one information for you. Long story of EPA from prostaglandin, especially from Philip Needleman. Prostaglandin story, PGI3 and thromboxane S3. That is a major mechanism of anti-thrombotic action of EPA. However, new insight in collaboration with incretin. 2 years ago, EPA receptor GPR120 in testing, finally production of GLP-1 increased. This compound, this hormone have cardioprotection. Further study is required, the combination of EPA and incretin. This slide, the last presentation of today's presentation. Finally, I express many thanks for many people. And finally, deepest thanks for Dr. Yon Daerbel. Thank you very much.